this video, let us talk about LE cell, LE bodies and LE cell phenomena. Coming to LE bodies. Anti-nuclear antibodies cannot penetrate the intact cell. What is an intact cell? Intact cell is a complete cell which has the nucleus and everything in it. Here the cell we are talking about is damaged DNA. So this anti-nuclear antibodies cannot penetrate the intact damaged DNA cell. But when the nuclei are exposed, this anti-nuclear antibodies fuse with the damaged DNA antigens. When the antibodies and the antigens fuse, it forms an antigen-antibody complex. This antigen-antibody complex which is formed is the LE body. So what is an LE body? It is a fusion of anti-nuclear antibody and the damaged cell nuclear antigen, right? The fusion of this both creates an LE body. Now it is also called as hematoxylin body. Why? Because it is basophilic and it absorbs the hematoxylin stain. Simple. So what is an LE cell? LE cell are the leukocytes, mainly this neutrophil and the macrophages, which engulf this LE body are the LE cell. So the neutrophils and the macrophages that engulfing the LE body is the LE cell. LE cells are the microscopic findings in the systemic lupus erythematosus, but not specific to SLE. This can also be found in pericardial and the pleural fluid, and also in case of rheumatoid arthritis, etc. Now coming to this picture, this first picture is a normal neutrophil. It has tri-lobed nucleus, and nucleus is placed centrally, and these are the granules. Now coming to the LE cell. So in the LE cell, what is an LE cell? It is a cell which engulfs the LE body. Middle portion, which you are able to see in violet color, is the LE body engulfed by the neutrophil. Now this neutrophil with the engulfed LE body is called as LE cell. What are the changes you can see in an LE cell? First thing is this trilobed nucleus is pushed peripherally because the central part is taken up by the LE body. Now coming to LE cell phenomena. The principle behind this phenomena is anti-nuclear antibodies fuse with damaged DNA nuclear antigen. That means antibody and the antigen fusion only occurs when the nuclei are exposed. The first slide we talked about first point, right? This antigen nuclear antibodies cannot penetrate the intact cell. So this is the mechanism here. Anti-nuclear antibody fusion occurs with the damaged DNA antigen only when the nuclei are exposed. So coming to the procedure, first we have to expose the nuclear antigen. So when it is exposed, this fusion occurs, right? So for the exposure, we need to do it by the rotatory bead method. When we use this rotatory bead method, by the continuous rotation by the glass bead, this nucleus get exposed. So when exposed, it forms antibody and the antigen complex. That means LE bodies. Now these LE bodies are engulfed by the LE cells. These LE cells are seen under the microscope. Commonly, these are the microscopic findings in case of SLE. So this is the first diagnostic test for the SLE historically. So this is about LE cell, LE bodies and LE cell phenomena. Bye.